So focused ultrasound has a potential of offering a very targeted and minimally invasive alternative to current neurostimulation strategies. The main issue is that the mechanisms underlying this method are still relatively unknown, which limits its application. So while numerous studies look into studying the safety and efficacy of this technique on in vivo models using complex repetitive pulse neurostimulation procedures, our approach was to go back to a relatively simple neural model and to study the effect of the fundamental building block of those repetitive pulse neurostimulation protocols, which is a single pulse. And so for that, we used a human progenitor cell line called RINCELVM, which was stained with a calcium-sensitive fluorescent marker to be able to assess the neural activity. And we used an inverted microscope coupled to a camera to record this activity on a large portion of the network. And so our approach was to study the biomechanisms of single pulse focused ultrasound neurostimulation and to find a set of stimulation parameters to allow for a high degree of repeatability and spatial temporal selectivity. So we did that by promoting the initiation of neural responses using acoustic cavitation force effects rather than cavitation effects, which are less controllable. And we also look into minimizing the size of the targeted region and using shorter pulses to increase the spatial temporal selectivity. And so we also introduced an alternative stimulation modality to be able to compare the outcome of the ultrasound stimulated responses with current descriptions of the effect of mechanical perturbations on cells. So we did single cell mechanical stimulations which allow for repeatable stimulations based on the deformation of a single cell membrane. So we deformed this single membrane using a patch-clamp micropipettes. And for the focused ultrasound stimulation setup, we used spherical transducers, which could be driven at frequencies ranging from 700 kHz to 8 MHz. So using that, we were able to apply mechanical perturbations on regions of varying sizes by changing the frequency of the stimulation. And so we also recorded the pressure fluctuations during the stimulations. And we saw that for pressure levels capable of inducing neural responses, the lower frequencies were more prone to initiate large-scale cavitation events, which could lead to cell damage and uncontrolled stimulations. And so we preferred to use higher frequencies to increase the spatial temporal selectivity and minimize the occurrence of cavitation. And so after stimulations, we saw a strong and sustained calcium elevations for cells located within the targeted region. And this was followed by the omnidirectional propagation of a calcium flux throughout the network. And so this was very repeatable, so to be able to compare multiple responses, we perform the segmentation of the images to be able to extract the fluorescence intensity variations of individual cells. And with that, we were able to perform the detection of the cell responses. And by using the center of the response as our reference, we were able to extract the spatial-temporal dynamic and distribution of the response. And with that data, we were able to evaluate quantitative parameters, such as the mean propagation velocity. And we saw that this specific parameter was actually similar for both focused ultrasound and mechanical stimulations. Then however, we can see that this parameter is quite limited, as it does not describe the response dynamic very accurately. As we can see that the propagation velocity is actually way faster at the beginning of the response and slows down over the course of the propagation. And so this is more like a diffusion process. And we can actually see that this dynamic also exists for the ultrasound stimulation. The only difference is that the fact that multiple neurons respond immediately after the response, the response distribution is more spread out. And so we can actually link the frequency of the stimulation with the size of this initially triggered cell cluster. And so we can see a clear relation. However, we can see that as we expect for the high frequencies, we can see that we have a cluster of cells which varies with regard with the stimulation frequency and the size of the mechanically perturbed region. But for the lower frequencies, we can see that we have the initiation of responses on smaller regions, which are still located within the targeted region and the displaced region. But we think that if you recall from previous slides, we think that the occurrence of large-scale cavitations is actually responsible for the creation of those responses, as we think that a small bubble, imploding bubble, could create mechanical perturbations capable of inducing those less controllable responses. And so if we go back to the response dynamic, we were curious what kind of signaling mechanism could be responsible for this diffusion dynamic. And we thought that it could be either the propagation of signaling mechanisms along the axons of the network, or the diffusion of a chemical compound in the extracellular media, which could trigger additional responses after the stimulation. And so we looked at the propagation velocity of the calcium flux along neurites or axons of the network. And we saw that the propagation velocity was actually pretty much constant, regardless of the orientation of the neurite or axon, compared to the propagation direction of the broad response. We also see that the propagation velocity is way faster than the mean propagation velocity we measured earlier. And so we think that this diffusion dynamic is actually due to the architecture of the network, and that it's actually a signaling mechanism occurring over the neurites of the network. And so with that, we can say that focused ultrasound is capable of evoking, via the radiation force, highly targeted calcium responses, which can later initiate network-wide signaling activity. So further work is still needed to continue the characterization of those mechanisms. However, we think that this approach should allow for better understanding and control of ultrasound neurostimulation. So with that, I would like to thank you for your attention. I would like to thank everyone involved in this project, and I will now take your questions. Thanks.